ourselves every day, how soon are we going to get a vaccine for coronavirus? Which country is going to be the first to produce it? I have a very special panel. Kiran Majumdar, the promoter of Bicon. She's really been at the forefront of tackling the COVID crisis, involved in a number of ways, both on the government front as well as her company. We also have Hasmukh Rawal of MyLab, which is at the forefront as far as testing is concerned. It's been in the news for its testing kits. We also have joining us a very special guest from Israel, Professor Gershoni from Tel Aviv University, who was recently granted a patent by the US for his coronavirus vaccine design. So thank you all very much for joining us. Kiran, if I can start with you, I read that the fastest it took for a vaccine to be produced was about 4 years. As far as the coronavirus vaccine is concerned, people are talking about 12 months, 18 months. If you have to give us a timeline today, what would it be? How soon can we see a vaccine?
So first and foremost, Chandra, thanks for having me on this panel. And I'd like to start by saying that, look, I think this pandemic has really been a rude wake-up call for the world. Now, if you actually think about the vaccine efforts in the past, and if only we had developed a SARS vaccine and a MERS vaccine, I can tell you that the world would have been in a much better place. Unfortunately, the vaccine development efforts for SARS were dropped when everyone thought that this is an Asian problem and not a world problem. And basically, it was just sort of, ignored thereafter. So it's very unfortunate that the coronavirus family was not looked at as a very potential, pandemic or a lethal kind of, or a virulent vaccine, a virulent strain. Now, what I would say is that when you look at what we are doing today is obviously we do need to abbreviate the timeline from the start to the finish. So I think we do need to make sure that we have abbreviated paths to see the vaccine in for people before too long. Now the problem is that there are problems and there are opportunities. I would say that there is enough technology, there is enough data analytics, there should be enough past experience to actually give us the comfort that certain data analytics or data is giving us the comfort of fast tracking the trial. So I think we do need to use a lot of post hoc data. We need to learn from some of those trials. And in future, I think we need to have a preparedness platform, a rapid response platform, such that we can actually develop a vaccine candidate very, very fast. But for this, you will need to have a surveillance mechanism for the virus, for various viruses for that matter, and which track its genetic evolution and which keep looking for new viruses, okay, in different families. So I think these are very important opportunities for global research and global partnership. Now, to come to the question of how soon will this vaccine reach people and can be, when can we immunize population with a COVID-19 vaccine? Well, as you know, there are 88 vaccine programs that the world is pursuing today, okay? And many of these have also entered the clinic. I think about 17 programs have sort of entered or just about entering the clinic. The most advanced, I would say, is the Sinovac vaccine from China, okay?
Right.
That at the moment appears to, at least in the animal studies where actually where monkeys were actually inoculated with the vaccine and where there was a challenge test done on the monkeys, it seems to be showing a lot of promise. I think people have started getting inoculated. And let's hope for the best. I don't know whether it will work or not. Finally, it all has to be proven in a clinical test. Then comes the effort that has been announced by the Oxford group.
That's right.
Also just entered the clinic, and let's hope that also gains traction, because they feel very comfortable that before the end of the year, they may be able to start immunizing people. Now, there are many such claims. I think, of course, the US is pursuing certain vanity projects because I think the kind of mRNA vaccines that are being developed, although there's a lot of money being thrown into it, I don't know how easily you can immunize large populations with a product that needs to be refrigerated at minus 80 degrees centigrade, which needs to be infused into people rather than jabbed. These are some of the things we need to look at. And for all of us in the world, it's about an affordable vaccine. It's an easy to use vaccine. We cannot be having very expensive and difficult to administer vaccines. So I think those are some of the questions we need to ask ourselves. But if you ask me, I think because there's such an urgency and demand for a vaccine getting to people as soon as possible, you might see a vaccine at least being approved before the end of the year.
Okay, before the end of the year sounds hopeful. Now,
and of course, there are many types of vaccines. I would also like to add by saying that even in India, we have many very interesting vaccine programs. For instance, Minvax is actually developing a vaccine based on a measles vector. They've basically taken a measles vaccine, they've taken the measles vector and then basically inserted the COVID-19 genetics into it and hoping that that will deliver the immune response that you want, because it's done very well for the measles vaccine. Then you have, a company called Seagull that is developing its vaccine based on virosomes. Then there are other companies that have developed CRISPR-based technologies to develop the immune backbone of the vaccine vector. So there's lots of efforts going on, but I'm sure John will be able to, talk a lot more about vaccine design. So I'll end here.
Right.
Indian vaccine could, again, if all goes well, it could be in the market in a year's time.
Yeah, in fact, that's what I was coming to. The scientist Gagandeep Gang, she recently said that at least half a dozen Indian companies are actively working on vaccines at this point. Minvac, you mentioned, then you have the likes of Serum Institute and Bharat Biotech and Biological E and others. So will we have a made in India vaccine? What are the chances of that happening before the end of the year?
Well, a made in India vaccine is definitely going to be a possibility because, as you know, Serum Institute has partnered with the Oxford group in terms of producing the vaccine. So probably you will have a made in India vaccine if all goes well in that particular trial, because they will be making the vaccines for sure, apart from other companies.
Right. Professor Gershoni, if I can come to you, you have been studying the structure of this vaccine, the structure of this virus, the design of this virus. I read a report recently where you said you were you had completed two-thirds into developing this vaccine. You were hopeful that it will happen soon. Can you tell us about the efforts that Israel is taking on this front, that you are taking on this front as a scientist?
Well, there are a number of efforts in Israel. I can't speak for all of them. One is from a laboratory up north in Migal that they're trying to adapt a vaccine based on a gamma coronavirus that's infected chickens and birds. However, our work is directed obviously to humans. So is theirs, but our experience has been SARS and MERS. And I agree totally with what Kiran had said previously, that it's unfortunate that much of the research that was done looking at SARS in the past and then MERS hadn't been brought to fruition. We started looking at the interaction between viruses, the virus spike protein and their corresponding receptors for many, many years. We've looked at different viruses such as HIV and flu, of course. And in 2004, we became interested in SARS coronavirus and identified what we believe is a very specific neutralizing epitope within the spike. It is the receptor binding motif. Those of us who are more familiar with the spike protein of coronaviruses, the carboxy terminal domain, the CTD, harbors what is referred to as the receptor binding domain. And that's about 200 amino acids. And this structure is responsible for interacting with the corresponding receptor. In the case of SARS, we're talking about ACE2. And it was our notion that looking at the atomic structure of this aspect of the spike protein, that we might be able to actually isolate out just the receptor binding motif, this very specific surface, which is about 50 amino acids long. The problem with this, and I'm sure most of the audience is aware of it, is that when you isolate out a small segment of a large protein, it's not sufficient to simply synthesize its amino acid sequence, but you have to make sure that this structure also folds into a functional physiological conformation, that it continues to look like the virus, if you will. And this was a major obstacle. And we developed a technology which allows us to reconstitute a functional receptor binding motif, the Achilles heel of the virus. And by the time we actually came to what looks like very good and encouraging results, as Kiran had said, nobody particularly cared, because SARS seemed to have petered out and it seemed to be uninteresting and no longer a threat. And so here we had a very good solution to a problem that nobody cared about. And this is unfortunate. But then when MERS evolved, we again examined the atomic structure of the spike protein of MERS, and were surprised to see that the critical components and the structures that were necessary and really the essence of our technology exist in MERS as well. It's a little more complicated, but for the most part, we saw many lines of similarities. So we immediately implemented everything we knew about SARS into MERS. And this was gratifying in the sense that we were able to reconstitute the MERS receptor binding motif. And again, this is a bit larger. It's about 70 amino acids. And here I want to also mention that I didn't do this by myself. I'm just a professor. I have colleagues that are doing the work. And actually, the person who did this very interesting and very good scientific project is a co-patriot of yours, Lakshmi Narasaya Uppalapati. He's a young Indian who's been working in my lab for many years, and he is truly excellent. And so it gives me pride to be able to participate here and mention an individual from Andhra Pradesh who contributed much to this project. And so we had our results on MERS, and as we were writing it up, all of a sudden COVID-19 appeared. And so I would say that we were very fortunate in the sense that we had 15 years studying coronaviruses behind us. And Lakshmi immediately recognized, looking at the genome and the DNA, well, actually the RNA, sequence of the virus, we could identify what appeared to be the candidate receptor binding motif. And we're currently working on this aggressively and making what I believe is very serious progress. And I would hope that in the course of the next month or two, we should have a fully reconstituted receptor binding motif. And if during this webinar I have the opportunity, I would explain why I believe that having a receptor binding motif could necessarily be very important to the coronavirus vaccine effort, because I think most of everybody has been focusing on the full-length spike. And whereas it contains exactly what we've been able to isolate, it also contains many surfaces that can elicit undesirable antibodies, and possibly we may encounter antibody-dependent enhancement, ADE, which has been reported both for SARS and MERS. And I think that the unique characteristic of the immunogen that we propose is that it can focus very clearly the immune response to precisely where you need it. I'm sorry, I can't hear you. Chandra, you are muted.
Oh, okay. Yeah.
Good.
Right. Thank you for that, Professor. And it's amazing that an Indian scientist working in your lab is possibly going to play a very big role in producing a vaccine for coronavirus. So he might
Without a
be
doubt.
working in Israel, but we are going to have an Indian origin scientist in Israel working on that.
Without a doubt.
That's fantastic news, something that all of us can take a lot of pride in. If I can come to you, Hasmukh, till the time we find a vaccine or the treatment, what's really going to be crucial is testing, contact tracing, quarantine, social distancing. That's what we have to do aggressively. MyLab has, got its testing kit approved. You're now talking about ramping up numbers in a big way, where you will be able to produce millions of kits in say a month or two to come. Take us through where India is with respect to testing compared to other countries in the world. There's always criticism that perhaps we are not testing enough. There's also, the testing infrastructure that we have. Hasmukh, can you take us through where we are as far as the testing is concerned?
So definitely, till the time any therapy or vaccine comes in place, the testing is going to help any of the countries in the globe to make sure that the disease are contained and the healthy are able to work. So I would definitely give precise on 3Ds, which definitely the whole globe should start following now is distancing, where you will be able to prevent the spread. Diagnostics, where you will be able to contain the spread, and deterrence with vaccines, where you will be able to prevent it for a long-term prevention. But by the time the vaccine is coming, I think diagnostic is going to play a major role. Now, when I talk about India, yes, I think there will be questions about India's diagnostic response till now, but I would like to give you some facts where I think the whole world should look at us as a country who is able to fight the disease compared to most of the developed world and so could not be able to do it. So just to give you an idea, from the date we have I think we are the first few countries in the globe who started doing diagnosis of the people who are coming from the infected areas. I think many of the countries failed there. From the time we have checked the first 100 corona positive person till date, we have increased the diagnostic capacities 100 times, more than 100x times. I think when we started, we were not even 100 country with the diagnostic capacity which compared to the globe, where or maybe per million people or total number of tests we have done. As we speak today, we are the 10th country in total number of testing done and preparing ourselves for more and more testing. Of course, looking at the population, demography, density, we definitely need more testing. But let me tell you, we are the 10th in the world in terms of total number of testing which are happening today. I think ICMR and Indian government has given a great response in fighting this epidemic. Just to tell you in terms of infrastructure, from 2 labs to more than 390 labs we have touched within last 1.5 months. That is a tremendous number. At the same time, let me tell you, we have been very careful about biosafety and the quality containment of the infrastructures where we are allowing the labs to be taken. Because let me tell you, the diagnostic though is important, making sure that the right labs with the right practices in place are only able to do the testing, because it may also be the reason for the spread, and that's what we have seen in many countries where the labs themselves become a reason for the spread. Testing locations, I think we started with 2 labs, and today we have more than 30 states and territories where the testings are being happened, and I think we are able to cover different geographies across India. The machineries have increased. The approval for the test, we are very sure that though slowly, but only the right kits are getting approved, so that the right solution is in place without any hurry, though despite all pressures of the globe or maybe India. We have tested more than 37 to 38 companies, and only 11 or 14 have passed as we speak today, compared to the globe where on emergency use only self-certification based companies are getting passed. I think India is very careful in allowing the right companies to go through the funnel, and only the right solution goes to the masses despite all the pressure on
right.
We have crossed more than 5 million plus tests, and that's a huge number. As far as the testing population is concerned, we have around 4% compared to WHO level, where anything about 10% positive rate is a area of concern. So I think India's response in terms of testing is fairly good. Though after the lockdown, I think that though the number will increase, I think that has given enough time for companies like us to make sure that we ramp up the capacity, we make sure that everything falls in place. Normally the testing, no matter whether you are in testing or in therapeutics or in vaccines or in PPEs or healthcare, you need some time to make sure that the enough inventory of the product is with you at place. That is around 28 to maybe 40 days time. I think the lockdown has given enough time for companies like us to prepare ourselves, make sure that the country doesn't need any outside support, and I think we internally can take care of our demand of it.
Right. Kiran, if I can come to you, we're starting to get questions from people who have logged into the webinar, and they want to know about Biocon's own efforts with respect to, finding a vaccine. If you are doing something like what global companies like Johnson & Johnson are doing, if you can take us through Biocon's own efforts as far as vaccine is concerned. This is a question that we've got from a person who's logged on to the webinar.
So, Biocon, of course, has many companies within its group, and one company is Syngene International, which of course is a research services company, and we are actually involved in a number of fronts. One is, of course, Hasmukh, MyLabs is actually, we are partnering with them to basically giving them some of the components that they need for their kits, because we realize that all these kits that were being made in India were actually depending a lot on imported components. So we said, let's indigenize a lot of these components and bring down the prices. So one of the things we're working on is to really make many of these components indigenously, so that companies like Hasmukh will actually benefit from indigenized component sourcing. Syngene is also developing serological tests, because I think serological tests are also very important, because PCR test is very important as the first diagnostic that you need to intervene with. But then very soon you'll also have to start using serological tests to basically, which is based both on antibody detection and antigen based serological tests. So we are doing that as well. And as John just mentioned, you need to basically be very specific about the spike protein that you're going to use to develop those tests. So in fact, Syngene is also developing spike protein in a large quantity so that you can do a lot of other things with it, because you will need it for many things. So Syngene is actually developing the spike protein. It is developing the serological tests based on that. We already have an ELISA kit ready for the serological test today, but we are now working with another company to develop a point of care lateral flow technology based kit, where you can actually then do it very rapidly. These are rapid tests, and it's a point of care test. So that's another area where we're looking at. Then Syngene has been working very closely with many vaccine companies, including Minvax, including Seagull, and a few other companies, providing them with support. They are all small companies, so they need a lot of research support in terms of, doing a lot of things for them. So I think that's how we are working. We are partnering with these vaccine companies and giving them the kind of support that they need to basically get it IND ready and getting it into the, sort of the preclinical. We also have animal models, so we are trying to help them with that, and then eventually into the clinic as well. So and we also have a clinical research arm which actually can help with many of these clinical trials, and we have done many vaccine trials in the past. So that is how we are actually working and partnering. And just yesterday, day before yesterday, we got Syngene's Syngene's research lab got converted into a diagnostic lab to offer tests. So that was another, because, they wanted to basically reach out to anybody who had PCR, sophisticated PCR infrastructure. We, of course, were one of them. So we offered to also enroll as a testing center. Of course, we are doing it free of cost, because I decided that, as a company, we should not be charging. We should be doing it out of our CSR. So we've just started testing. So that's another thing that we're doing. So multiple things, I would say.
Right. Thank you for that. Professor Gershoni, if I can come to you, I want you to tell us about, the life cycle and the development of a vaccine. I read somewhere that, apart from animal trials, you'll have to go through 3 phases of human trials. The first trial where you have about 20 people, the second phase where you expand it to 200 people, and finally the third phase where you expand it to between 5,000 to 100,000 people. Now, considering we are in mission mode, we are in the middle of this pandemic, will this normal life cycle apply? Is there some way we can ramp up the development of vaccine in terms of the time it will take, in terms of this entire life cycle that it will take? Take us through that. I mean, are there shortcuts that we can take without compromising the vaccine?
Well, first of all, I'm sure our panelists have as much experience and probably more than me, but nonetheless, I can walk through the general paradigm of how one develops a vaccine and suggest possible corners to cut, if you will, because as we've all indicated, there's a sincere sense of urgency, and anything we can do to possibly get a viable prophylactic protective vaccine into somebody's arm is worthwhile. So I think that there's a tendency to possibly use some of the platforms that other companies have already developed, J&J and Sanofi and others in India, various platforms for vaccines, and have a greater willingness to go directly into phase 1 clinical trials into people. So I think that the amount of animal testing it's worthwhile and necessary, but I think this is an area that people are willing to say, well, let's try to go for it. Let's see if we can at least evaluate toxicity, which would be your phase 1. Dosage in phase 2 is also something that is not too demanding. So I think that the major challenge is what would be required of a phase 3 clinical trial in order to show efficacy. Here, actually, if we're willing to be a little more creative, I think we can possibly cut corners. From what we understand amongst the many now unfortunate examples around the world, there are almost 3 million cases thus far of COVID-19. It appears that younger individuals, younger than myself, are actually pretty resilient to the disease, that it's not a horrific disease and not terribly life-threatening, especially for young individuals, young adults, maybe at the age of 20 or so. So I don't find it objectionable. I understand that it's considered by some as unethical, but let's put ethics aside for a moment. I don't want to be necessarily then labeled or tagged the Israeli is unethical, but let's put politics aside as well. And let's just try to be open-minded and figure out, would it be unheard of to identify a small group, maybe a few hundred volunteers, and one could possibly immunize them, vaccinate them. I think we have enough knowledge in order to then evaluate their serological response. Have they been able to mount a significant antibody response? And then we can also test to see the quality of that antibody response in vitro. Does it seem to be neutralizing virus and so on? And so this is something that could be done rather quickly. I mean, we can't speed up our immune response, but if we were to take maybe 100, 200 volunteers and vaccinate them, and then subsequently identify those that have a good serological response, a high titer and a neutralizing titer, then I personally don't think it would be outrageous to challenge them with bona fide virus. Now, the worst thing, of course, that could happen is somebody could die. So I'm not belittling this. But in reality, the experience that has been gained over these last 3 months is the chances of a young, healthy individual with no complications probably would withstand the disease if it turns out that the vaccine protocol was not protective. Now, obviously, what we're expecting is that if we've vaccinated an individual, he's gained a strong, robust, serological neutralizing response, we believe that he should also be protected. And therefore, this is something that if we did a volunteer set of individuals, vaccinate them and challenge them, it runs against everything we've ever considered as being ethical. We know way back that Jenner did this with James Phipps in smallpox. And so that was outrageous. But I think today in the 21st century, and thinking about the horrific consequences of the pandemic thus far, it would not be outrageous to carefully consider a vaccination and challenge of a group of volunteers. This would probably be the greatest step in trying to cut corners. And again, I know that it would be immediately objectionable by many groups, but it could be possibly beneficial. And so I leave you with that thought. Obviously, I understand the concerns, but I would at least like to point out the possible advantages.
Right. Even as we speak, we, Adar Poonawalla of Serum Institute has just been talking to our channel, and he said that he's hopeful that the Oxford University vaccine will be ready by October if human trials are successful, and clinical trials in India, he says, will begin in 2 weeks, and the COVID-19 vaccine will cost about 50,000. That's coming from Adar Poonawalla. He just spoke to our channel a while back. But Hasmukh, a lot of questions coming your way on the capacity. How many testing kits do we currently have in the country? What is our capacity? How much has been supplied? Is there an umbrella figure that you can share? Since when it comes to testing kits and testing your
surely testing is needed, and definitely we need to produce more and more our country. And I think not only MyLab alone, but most of the players inside the country and outside should support getting testing done till the time we have a reliable vaccine coming. I would like to correct one figure is not a 500,000, but not 5 million, but more than 500,000 tests have been done by our country till date. To be precise, 6.7 lakh, 670K tests have been done. To tell you a capacity, as we speak today, we have supplied more than 350K tests. By this week, we will supply, so last 4 weeks, we have made total 350K tests. This week, we will make single week alone, we'll make 350K tests in this single week. And by the first week of May, we'll be producing more than, every day we'll make around 200K tests. So that is the minimum we would be producing. And of course, that is with the help of Mr. Adar Poonawalla as well as partners like Syngene. I think we will be in well-versed position to support a country's inherent requirement. Looking at the ICMR number as well, a country is also planning to test as good as 100K tests, 1 lakh tests a day. And I think from there, they would like to ramp it up. I think we will be in position before 15th May, we'll be in position to support the whole country's need for next 3 to 4 months as well.
Okay, so you'll be in a position to support the whole country's needs in the next 3 to 4 months. Is that your timeline, or in the next month?
By coming 2 weeks only, we will be able to support for next 3 months. So total 3 months.
For the next 3 months. Super. Okay. Kiran, a lot of questions coming your way on vaccine, COVID vaccine, what Bicon is doing. You've already answered that. But people are also concerned that we are still in the very early stage. WHO has given has sort of cautioned that it will not be such a good idea to issue immunity passports or immunity certificates, because there is a chance that this might be a recurring phenomenon, that this immunity will not be permanent. So how will this vaccine pan out? Is it something that people have to take periodic doses of? People are also talking about different kinds of vaccines, DNA vaccine and mRNA vaccines and vector vaccines. What do you see on this front?
Well, I think, every vaccine has to be evaluated for, how long it is effective. I mean, these are things that all vaccines have to be evaluated for. That's why I think you need to extrapolate some of that data as to, what is the immune response? What is the antibody titer? What is the neutralizing antibody titer? How long is it remaining in your body? That's why you can't short circuit it too much, because you have to actually evaluate all these things and decide whether you need to be vaccinated annually, whether you need to be vaccinated once in 2 years, 5 years, 10 years. This is all data that will come out finally. But right now, even if you can get data to show that, there is a effect for a long period of time, that is good enough to say, okay, you can vaccinate once a year or whatever it is. But before you can get all this data, it's very difficult to speculate, Chandra. So I think talk about immunity passes, it'll all depend on how long is the, what is the duration of the effect of the vaccine. That is what you'll have to see.
Right, right. And in terms of the types of vaccines, Kiran, people are talking about vector vaccines and DNA vaccines.
No.
And
there are lots of types of vaccines. I mean, there are live attenuated vaccines, there are DNA vaccines, there are RNA vaccines, there are virosomal vaccines, there are umpteen number of vector-based vaccines. So I think, it all depends on which is going to be easy, which is going to make it first, which is going to really be effective. What does each vaccine do? For instance, even if I mentioned that the mRNA vaccine is very, very complex and very difficult to deliver, but if it gives you a 5-year effect of immunity, then it becomes a very valuable vaccine, right? But if it's going to do just the same as other vaccines, then it doesn't have the same kind of, impact. So I'm just saying each vaccine will have its own kind of pluses and minuses. So you need to evaluate these vaccines. I think each vaccine will have a good opportunity to play, because the world will really need, and I can tell you post COVID-19, I think the human race will want to really immunize themselves against almost anything and everything. I mean, that's look at it. So I just think that, this particular wake-up call has told us that, don't take things so carelessly, because I know I've been very, very regular about my flu shots. And I know many of my friends don't take a flu shot, saying, what's the point? But now I know everyone will want to take a flu shot. Then, of course, once this vaccine comes out, I'm sure everybody will want this vaccine for sure. Okay, so you're going to have 8 billion people around the world who need to be vaccinated with this with this vaccine. And you know that, so therefore I believe that many vaccines have an opportunity to play in the market. It's not just one vaccine. But obviously the first vaccine will be something that everybody will want to use. The first approved vaccine, there'll be a rush. But hopefully you'll get a spate of vaccines coming out over the next 1 year, and then you'll probably be able to then feel much, much safer and more secure about the pandemic. And so these are the kind of things that you need to really look at.
Right. We're just approaching the final 5 minutes. Professor Gershoni, a question to you. This came from one of my friends. He says, assume there is another pandemic in 2 years. Will the efforts we are putting in today help in an accelerated development of the next vaccine? Will it help in crunching the time cycles if, God forbid, we have another pandemic, another random event like this 2 years down the line?
Well, I would like to address 2 points, because what you're suggesting is, God forbid, we have a totally different virus, a new pandemic. Then, as we all know, vaccines are extremely specific to the pathogen. And so experience gained on MERS does not promise a better vaccine for COVID-19. But an underlying question that has been mentioned and alluded to is the fact that how confident are we that the efforts being made today are going to be relevant to next year's version of COVID-19? And people, of course, take the example of flu, that we need seasonal flu shots. I think here we actually, on a sense of optimism, we can gain some confidence here, because COVID-19 virus is a coronavirus, and coronaviruses are known for being exceptionally large RNA viruses. And because of that, they've evolved very uniquely to have an RNA editing and repair system. So they tend to be rather stable. So I don't think that we're going to be confronted with a very good vaccine this year, that next season will be outdated. I think that coronaviruses generally are relatively stable, and therefore I expect that the vaccines to be developed today are going to be useful next year. Now, another thing that when you had mentioned the different types of vaccines, I think regardless of what type, there will be need also for a boost system. I don't think that a primary vaccination is going to be sufficient. And here's where I believe that we may actually have something to contribute. Because irrespective of the platform, if people are using Spike, then I would suggest possibly coming in with an RBM boost in order to specifically accentuate and enhance the antibody response to the receptor binding surface of the spike. And so we may see a combination, a prime with one and then an RBM boost on the other. And so I think that this is something to consider.
Right. On that note, thank you very much, Kiran Majumdar, Professor Gershoni, as well as Hasmukh Rawal for joining us in this special webinar. We've been getting a lot of questions, but I think our panelists took some of those questions on, testing kits, ramping up production, as well as vaccines. So thank you all very much for joining this webinar. And we hope it helped you answer the question that we ask ourselves every day. How soon can we get a vaccine? Hopefully it will be soon. And hopefully we will also have a made in India vaccine very soon by the end of the year. On that note, thank you very much for joining ET Now for this special.
Thank you.
Thank you.
And definitely we will have India to produce the vaccine for the masses of the world. Definitely.
Just like we have MyLab producing the testing kit.
Yeah, sure. I have definite confidence that India will play a major role in vaccine as well.
Thank you.
Thank you, Hasmukh.
Thank you.
Thank you, Kiran. Thank you, Professor.
John.